barcoding. DNA barcode is an emerging technology that can be used in various fields such as species classification and food identification. It uses short DNA sequences to identify species. Traditional morphological identification features will cause identification errors due to homogeneity and variation. Compared with traditional classification methods, DNA barcode technology expands the scope of testing samples. Even if part of a sample is damaged, it will not affect identification results. DNA barcoding is able to put aside the illusion of similar morphology and identify species that are difficult to distinguish by traditional classification methods at the genetic level. The establishment of a DNA barcode database can not only identify a large number of species at once, but also provide clear information about each species. It can not only make up for the lack of description of species morphology in traditional taxonomy, but also speed up the identification of known species. For next semester, I'm going to work inside lab and collect samples from to identify the salmon species. In recent years, reports on food safety issues such as food alteration and counterfeiting have continued to appear. Products and labels do not match. Meat alteration, shoddy goods, and other commercial fraud problems affect people's interest and health. Food fraud isn't a new phenomenon. Recently, the problem of food alteration and fraud has been paid more and more attention, and traditional detection methods can no longer meet the needs of food identification. DNA barcode technology is to identify food at the molecular level, which makes up for the shortcomings of traditional identification methods. Its accuracy, efficiency, and simplicity have brought a new revolution in the field of food identification. I can't wait to start my research on identifying salmon species from different restaurants next semester, and I'm so excited to be inside the lab. Thank you all for being here and listening to my presentation.
Okay, that was our presentation from Peiham Huang. Unfortunately, she couldn't be here to present live. Is Morgan back?
All right. And my presentation is on the basics of DNA barcoding and also providing the basis for my research project that I will be continuing in the upcoming semesters. So first we need to know, what is DNA barcoding? DNA barcodes are short segments of a gene. They're around 650 to 800 nucleotides long. In order for a DNA segment to function as a barcode, it needs to be present in a large number of organisms. For example, if we want to identify a species of plant, we would use a gene that codes for a protein involved in photosynthesis. If we wanted to identify an animal, we would use a gene that codes for a protein involved in cellular respiration. If we look at genes that are found in all living organisms, we can see that there's sections within those genes that are unique or variable from each species. And with DNA barcoding, we isolate the DNA from our sample, amplify the region in the gene that's variable, and then we can sequence it. And then based on that sequence analysis, we can determine the species of the organism by comparing it to known species. So a couple methods of DNA barcoding are DNA sequencing, which can be used to identify samples by comparing it to known sequences. There's gel electrophoresis, which provides proof of product. We add CyberSafe to the gel to stain the DNA in order to be able to view it under a UV light. When we run the gel, we can compare the bands to the allele ladder to prove the presence of the barcode. And then there's DNA isolation. After collecting a sample, the next step is to isolate the DNA from the sample. And first, we break up the tissue by adding lysis solution in the sample, and then we can isolate the sample by using PCR. All right. And then for my future project, my study site will be local forests and parks around the Raleigh-Wake Forest area. The protocols I will use are DNA extraction, amplification, proof of product, and sampling and sequence analysis, which are the methods that I discussed on the previous slide. And you can easily identify my sample species, which is the kudzu vine, based on the rapid growth, the climbing nature, and the invasive habit. And you can see some pictures there on the left. It's very common around this area, and you've probably seen it on the sides of highways, in forests, local parks. It's very common around here. All right, so my research questions that I'll be answering in my project are: how is the invasive species Fuaria montana, or the kudzu vine, different from other native North Carolina plant species? How can we identify and predict places it grows and slow the spread? This research is important in order to slow the spread of invasive species, and the first step is being able to differentiate between native and invasive species. An invasion of kudzu in an area threatens ecosystems and leads to decreased biodiversity. Kudzu was introduced in the US in 1876 at the Philadelphia Centennial Exposition, where it was promoted as an ornamental plant and a crop that was also used to reduce soil erosion. Kudzu kills other plants by suffocating them under their leaves and blocking off access to sunlight, or can even crush them under their weight. They can grow up to 100 feet in length and even weigh up to 400 pounds. And once established in an area, kudzu expands rapidly at rates up to a foot each day. It's also very flexible and adaptable to many conditions and can take over areas where native plants can't survive. And it's really important that we know how to identify this species so that we can slow the spread and therefore maintain a good level of biodiversity, and it won't be able to take over the native plants in the area. Just a couple fun facts, well, not so fun facts. It's known as the vine that ate the South because it's really common in areas from like Virginia all the way down the east coast of Florida and Texas, Louisiana, Mississippi, areas like that. So it's known as the vine that ate the South. It's also known as the mile-a-minute vine just because it grows so rapidly and it's hard to control. All right, and that's the end of my presentation. Big thank you to my mentor, Dr. Walsh, and for all of you here watching my presentation. So thank you.
Thank you, Morgan.
Yeah, thank you. Sorry about the technical difficulties.
It happens. That's what we're here for. We have 5 minutes left in the session if there are any questions.
I have a question for Morgan. This is Dr. Littlefield. Morgan, first of all, I want to congratulate you, and you say that you did a great job on your presentation, and I'm so excited that you're going to be doing DNA barcoding research, as it's certainly a passion of mine, along with Dr. Walsh and Scott Johnson. So part of my question is, as you've been researching this, are there species of plants, either native or other invasive species, that look an awful lot like kudzu that makes it sort of difficult to identify the kudzu based on site or morphology?
There's a lot of vines that are similar, but kudzu is typically easily able to be identified because it grows really fast.
Right.
And it grows really fast. It climbs up trees. I don't know if you saw in the picture. It's super common around this area. You can see it growing, like it grows on power lines, on trees, and it like grows up. So it like looks really weird, and it's easy to identify based on other vines that typically like start at the top and then grow down.
Right. I guess the question that I'm kind of trying to get you to think about is, if you're going to be going through the whole process of doing the molecular analysis here, one thing that's going to make your project stronger is if you're able to kind of say, this is something that can be difficult to identify based on site alone. And that's why it's really helpful to do the DNA barcoding, because it can be easily confused with this other vine that grows nearby or looks very similar. And that kind of helps to give you that extra kind of oomph with your research in terms of saying this is why DNA barcoding makes this so helpful, is because, you know, we might confuse it with something else. So that might be something to think about as you're moving forward with your research, just to kind of give it that context of kind of saying this is why DNA barcoding is so helpful with this, because it might be something that is hard for us to identify if we're just sort of out in the wild and looking at it. Does that make sense?
It does. Thank you.
Yeah.
That's definitely like the direction I want to take with like my future research is how can we use DNA barcoding to verify that this is the species we're looking for, because it's a very like dangerous invasive species. It's dangerous to native species. So it's very important that we know how to identify it so that we can slow the spread and ensure the biodiversity in the local areas.
Right, absolutely. And I think that context is great. So I think like as you move forward, you might actually be able to, as you learn more about it, kind of say, oh, well, one thing is that it grows in this area with this other vine that's native and totally fine. So we don't want to get rid of this vine. We want to get rid of the kudzu, and DNA barcoding can help us figure that out. So I think that's something that'll just make your project even stronger. I think it's a great idea. So again, really great job. Nice presentation.
Thank you so much.
Anybody else have any other questions? We've got 2 minutes remaining for Morgan or Amelia. Okay, well, I'd like to thank everybody for coming today. There is a link to the START program if you'd like to look at the next couple of sessions in group 2. And I am also trying to share a few links with you for, oh boy, that pasted really strangely. Some programs, the Futures program, the NC Science Festival, and a link to our START program if you're interested in checking those out. I'll try to make that look better. Last call for any questions? Okay, thank you all so much for coming. Enjoy your day.